Hi, this is Rethik, your biology subject matter expert from MemoNeet, and once again, welcome to the biology series. So, in this video lecture, we will be concentrating on another chapter that is molecular basis of inheritance, where we will be discussing about the Human Genome Project. So, what is Human Genome Project, and what are the goals, objectives of Human Genome Project, and significance, advantages of Human Genome Project? That is what we are going to concentrate here. Why? Because it's one of the most important and generalized topic. So we cannot miss out this specific topic, as there are DNA applications, there are PCR applications. So yeah, questions might come from this specific topic, from this molecular, biology. That's why we will be concentrating this topic first. So we all know that the DNA that actually determines what? The genetic information of a living organisms. So in another word, if I need to concentrate, or if we need to think of as whatever the genetic makeup, right, of an organisms or of an individual, that lies in the DNA sequence. So that means DNA is what responsible for one's genetic information. So if two individuals are actually differ, like if the two individuals, if they are differ, that means what? The DNA sequence should also be different, right? Say, like if you are different from your friend, that means also there has to be some DNA sequence which should be different, at least in some of the places. If you are comparing yourself with your brothers, relatives, whatever it is, you are feeling your physical appearance, your characters, everything is different, right? So that means even the DNA sequence should also be different, and at least at some points there will be difference, even in twins. So these assumptions led to the queries, that is question of finding the complete DNA sequence of human genome. So that means in a human genome or human gene. So what is the DNA sequence and how many genomes are present in human gene? So that was what the major question or query that came into the mind. So with the establishment of this genetic engineering techniques, with the time being, okay, when this biotechnology got advanced, small biology got advanced, so an introduction of this genetic engineering techniques got viral up. So with its possibilities, it became a possible to isolate and clone any pieces of DNA. So for say, like if you are having only this single, a small piece of DNA, you can easily isolate the DNA. You can clone the DNA to number of pieces that you want by a very, simple and very fast technique of determining the DNA sequence. And in that way, a very ambitious project that was launched, or that came out. So that is the sequencing what? Human genome was launched in 1990. So this is very important. In 1990, this Human Genome Project was launched. So now, what is this Human Genome Project? In shortcut, they'll also be calling this as HGP. Okay. So yeah, what is this Human Genome Project here? So Human Genome Project is actually a mega project. Mega is nothing but a large project. It has taken a lot amount of time actually. Okay. So I can call that as a mega project. This is one point. Then the total estimation, that is the cost. Okay. Like whenever you are taking up any projects, when you are making a plan or when you are doing some projects, right? Obviously an estimation of amount is required. So they have estimated the total cost for the project was approximately 9 billion US dollar. Okay, which is really very costly, and it went out many years project. So further obtained a sequence where they have to be stored in a type of a book form, and each pages of the book contains around 1,000 letters, and each book contains 1,000 pages. Then 3,300 such books were required to store the information of the DNA sequence from a single human cell. You're getting? So it is an estimation that how DNA sequence could be a complicator of storing the information. That means enormous information is there. That is what they are trying to say. That if you want to, if you take a single human cell and if you start sequencing it, you require how much? 3,300 such books, like a book having 1,000 pages. Okay, and in each page you will be covering what? 1,000 letters of informations will be stored. So that much a human genome will be. That is what they're trying to convey here. So this enormous amount of data expected to be generated also, that has to be like how we will be generating that much of enormous data by using a high speed computerization device for data storage and analysis of that. So that is the major one important thing. So there comes this Human Genome Project was closely associated with the rapid development of new area in the bioinformatics. So there comes this called bioinformatics. Okay. So in bioinformatics, you will be using a computer analysis to store up the large, information, gene information, genomic information, and all. So then, what are the goals of this Human Genome Project? So one thing is to identify approximately 20 to 25,000 genes in a human DNA. That was the very first goal. So next, to determine the sequence of 3 billion chemical base pair that make up the DNA, okay, human DNA. Then whatever the information that was available or we got, right, so that has to be stored in a database, and improve tools for data analysis. How do you analysis a data? So by improving the tool to analysis the data, that means easily if you have some certain, if you get some kind of new, nucleotides or base pairs, whatever it is. So having this, you can compare that with some, another nucleotides or base pairs, or if you have a protein, you can compare with another protein, pre-existing protein. That is what then transfer of this related technologies to other sectors such as industries and all, so that they could be utilized. Then we need to also concentrate very important thing, that is they had to be ethical and legal and social issues that may arise from the project. That means while doing this project, there should be ethical values should also be there, and it should be very legal, and it should overcome the social issues. So ELSI is nothing but ethical, legal, and social issues. So this project should arise or overcome all these things. Okay, so that was the major goal of this Human Genome Project. So how many years the project took? Approximately, they say, right, 13 years of project it was, and it was been coordinated by US Department of Energy and National Institution of Health. And during the early years of this Human Genome Project, initial years of this project, the welcome test, that is which is present in UK. So they became a major partner, and additional contribution was from Japan, France, Germany, China, and other countries also. So then they say like the project got completed in 2003, which is there in your textbook. So, what are the methods they have taken to analyze this Human Genome Project? Is the two different methods and significant methods that was been used. First is expressed sequence tag, wherein the genes were different, differentiated into the one apart, forming a part of the genome, and the other which expresses the RNAs. Then the second one is sequence annotations, wherein the entire genome was first sequenced and functional tags were assigned later. So it is nothing but first thing is we need to identify whether the genes, all the genes were differentiated into one another apart from, single. Then on which gene expresses what kind of RNA. So that is what they were trying to analyze was the first thing. Then the second thing is they first try to differentiate the entire genome. So they try to study the entire genome as first thing, and later their function, individual genes function, was later studied. So what is the process? What exactly the process of Human Genome Project was? So first thing is a complete gene set was isolated from a cell. So from one cell, a complete set of gene was isolated. Then it was split into smaller fragments. Okay, then the DNA structure was then amplified with the help of vector. Amplification is nothing but if you have one set of DNA here, so you can amplify into more set of DNA, something like that. So it was been amplified with the help of vector. So vector that was basically used is BAC, that is bacterial artificial chromosome, and YAC, that is yeast artificial chromosome. So these are the vector which is majorly used. You need to remember this. Then the smaller fragments, which was obtained were then sequenced with this DNA sequencing. And on the basis of overlapping region, what happens? The sequence were then arranged. Then all the information of the genome sequence were then stored in the computer-based program. Then this, where the entire genome was sequenced and stored as a genome database in the computer. So genome mapping was actually the major goal. Like we had to map, okay, on which particular space of chromosome. So what kind of gene? So what gene is actually present? That was the major goal, okay, which was to be achieved with the help of this microsatellites, that is repetitive DNA sequences. So they say a silent futures of the human genome project is. So what they got an outcome. Okay, one thing is the human genomes, right? They contain how much? 3,164.7 million base space, the length I'm talking about. So that is the base space. Then the average gene consists of 3,000 bases, but size vary from 1 to 1. Okay, with a large non-human gene being uh, the stropin at 2.4 million bases. That means the total number of gene is estimated all over in a human genome project. They have been estimated around 30,000, much lower than previously estimated 80,000 to 1 like lakh 40,000 genes. Then almost 99.9% nucleotide bases are exactly the same in all the people. So that's where they say that after the result, after analyzing the human genome project, they got to know that 99.9%, all the living human beings, they're sharing the similar nucleotide base. So that is what makes us connected. Then the functions are unknown for over 50% of the discovered gene, and less than 2% of the gene codes for protein. Okay, that is what we know. And the repeated sequences make up very large protein in the human genome. And they are stretches of the DNA sequence that are repeated many times. Some of the sequence, they have been repeated many times. So say like it have been repeated more than 100 times or 1,000 times. So they are though to have no direct coding function, but they shed light on a kind of chromosomal structure, how they might have got evolved because they've been repeating again and again. So that dynamic structure and all. Like chromosome 1 has most gene, so that is it has 2,968 kind of genes, and Y has the very least gene here, like 231 gene, 231, something like that. So scientists have also identified about 1.4 million locations. Okay. Locations are nothing but place where you can find them in a single DNA differences, like in one single DNA strand or differences. Yeah, they have found like 1.4 million locations. Okay, was been found occur in a human. This information promised to, bring up a kind of revolution or in the process of finding the chromosomal locations for disease-associated sequences and tracking the human history. Like once you know the location, exact location of the gene, okay, and the protein from which the gene is synthesizing, and if there is any disease which is related to genetic disorder or protein because of dysfunction of the protein synthesis, so we can work on that. We can know whether it is actually caused by this specific gene or not, just by knowing that location on the chromosome. So the applications and the future challenges that came up with this human genome project is, so one thing is we were able to have a clear cut clarity of the DNA sequence, uh, and that will help in the research, okay, through coming decades, leading to understand the biological system of a living organism. Then it also ensures a task will require that experiences and creativity of tens of thousands of scientists for various disciplinary in both the public and private sectors worldwide. And one of the greatest impact of having this human genome project sequence was enabling a radically new approach to biological research. By understanding the sequences and all, a new biological research came front. So in the past research studies, one or few genes at the time, like say like there were very less knowledge about genes and its specific role. But because of this sequencing, human genome project sequencing, one were able to, get to know that, okay, still new subjects, new, revolution came in research. So with whole genome sequence and new high, through technologies by using, we can approach a questions simultaneously and one much broad scale. We can start analyzing them. So they can also help in studying all the genes in the genome For example, all the transcripts in a particular tissues or organs or tumor, or how tens or thousands of genes and like the proteins work together interconnected with each other, and what are its chemistry of life, how they are coordinating with each other, everything will be able to understand by this human genome project. Okay, next. So this was all about human genome project. So after human genome project, there is another concept called DNA fingerprinting. So what do you mean by DNA fingerprinting? As I have already discussed, and we got to know that 99.9% of the base pairs, they are very similar among the human beings, right? So if one aims to find out the genetical difference between the two individuals or among individual of a population, what happens? The sequencing the DNA every time would be something time lapse, like you need to bring two individual at a time and you have to compare, you have to take that DNA and need to sequence one by one, so it becomes a lengthy task. So imagine like trying to compare two sets of the entire sequence. Okay, I'm talking about the entire sequence here, and you have a two set of people now, like you have a person A and person B, and you have to compare the entire sequence. It becomes a very time lagging one, and it will also take up a lot of money, manpower sometimes and all. So therefore, this DNA fingerprinting comes in that place where it becomes risky. We can take up this DNA fingerprinting technique, because if you have just a small little bit of any sample of an individual, you can easily study the differences of the base space or nucleotide, whatever it is. So DNA fingerprinting is one of the most promising in the forensic techniques using to identify an individual based on their unique genetic profiling. Okay. As I told, even though the sequencing is same, but there will be slight differences in the nucleotide position, on the DNA between the two individuals. So that is what we are going to concentrate on. So DNA fingerprinting involves what? Dif- Differentiating or identifying the differences in some specific regions in the DNA sequence. So we normally call those regions as what? Repetitive DNA. Since what happens, you can actually collect the DNA, right? So it could be like from where all possibilities you can collect. It could be from blood or hair follicles or skin, bone, saliva, sperms, and many other places. So from our individual, what happens? They will be showing the same degree of polymorphism, similarities. So they became very useful identification tool. If there is any slight, because similarities, say like you are in some specific spot where a crime had happened and you got some samples, okay, either it could be a blood sample. So now you have, say like on that specific spot, you have around 15 people. Okay, and there is only one person who might have done that crime. So what you will do is you are going to collect the blood sample which you have got, and you are going to sequence it with all those 15%. Since the sequences, the genetic sequence, the DNA sequence is very similar in an individual. Like my sequence, wherever you have taken the sample, obviously my DNA, will be similar. Okay, when compared to the other person's DNA. So that becomes a very easy way of, identifying who is the real culprit. Or it could be, to investigate who is actually a father or mother of the child. So I get to know the advantages. Okay, so this is one way of identifying. So the technique of DNA fingerprinting was actually, was developed by Jeffries. So the basic thing is he used a satellite DNA as a probe. Probe is nothing but the one which goes and attached to the one base pair, single base, something like that. So as a higher degree of polymorphism. So it was actually called as VNTR probes. That is nothing but variable number of tandem repeats. Okay. So the technique was used earlier involving southern blot hybridization technique, where we normally use this variable number of tandem repeats as a probe here. So what are the steps that involve in the DNA isolation? One is isolation of the DNA. So what is a major step of the DNA gel electrophoresis is So one thing is you're going to isolate the DNA. So from where you're going to isolate the DNA here? So you are going to isolate the DNA from the samples. I mean, it could be through the blood, or it could be through the skin, it could be through bone marrow, whatever it is. So now you are going to isolate the DNA. So DNA has been isolated. If you have a very less amount of DNA, it could undergo PCR technique, okay, polymerase chain reaction technique, where if you have a very less amount of DNA, so that will multiply it like into more number of DNA. So anyhow, now you have isolated the DNA. So now the isolated DNA, say like you have isolated a DNA. So by using restriction endonuclease. So what is the major function of restriction endonuclease? It acts as a scissor now. Okay. So it is going to cut the DNA into smaller pieces of fragments. So now we are going to run the gel electrophoresis. How? So say like this is the well. Okay. So these are the wells. So you are going to load the sample in the well, inside the well. So what are this? They are the buffers, and this is the gel. Okay. So now we are going to load. So what is the charge of the DNA? DNA charge is negative, right? So what happens at the one end of this gel electrophoresis will be the electrode will be carried where you are going to load the sample, and to the opposite you will be having another electrode which is positive. Okay. So at one end you will be seeing the cathode, and one end you will be seeing what? Anode. So now what happens, as the charges will obviously attract to the opposite one, and you will start to run the gel electrophoresis. So as a result, what happens? So obviously it will start moving in opposite direction, right? So to the direction where you have loaded, from that to opposite direction it will start moving. The smaller fragment, they will move first, and the larger fragments, they'll remain there itself, or they'll move slightly somewhat. Okay. So once this is done, the electrophoresis, it has been stopped. Then you will get different bands. Okay. So those bands, that the entire gel will be transferred to a membrane now. So we normally call either the synthetic membrane, such as nitrocellular or nylon membrane. Okay. So now if this is a nylon membrane, so this the gel has been transferred there. So now as a result, what happened? To this, you're going to add what? VNTP props, that is variable number of tandem repeats. So it is an hybridization. Okay. You are going to hybridize, like that means you are going to leave it for a time so that this props, will go and it is going to attach to the specific binding sites. Okay. In that way, we will be able to identify. So once this hybridization is completed, this V and T are props, they will go and they're going to hybridize it at its specific binding site. And later on, we are going to determine this hybridized DNA. So what is the amount of hybridized DNA by using autoradiography? So in autoradiography, if this V and T R props have been hybridized more with the DNA, that means this a specific person's sample is actually matching with the culprit one. So in that way, we will come to a conclusion that, okay, this suspected crime might be done by this specific person. This is how we come to a conclusion. And this is how we are going to estimate, not only with the crime, even with many other advantages. So this is how it happens. Like you have a DNA, when put this prop, obviously it is going to attain its position. Now, so one very best way of understanding what are the advantages are the forensic science. Obviously, you have a sample, and by using that, we can solve crime cases, murder, rape cases, and all. Next, we have paternity and maternity determination, whether the child belongs to whom. So that cases can also be solved. Then personal identification, like it can be used as to identify, genetic barcode to prior individual. So who is that individual, something like that. And even you can also use this to, diagnose this inherited diseases, such as it could be Huntington disease, hemophilia, fibrosis, cystic fibrosis, or, Alzheimer's diseases, sickle cell anemia, and many other diseases can also be identified in newborn babies. Then development of for you can also develop a kind of cure to for the inheritance diseases, okay, by seeing the history of some particular disorder, DNA identifying that, okay, this person might be having the disorder. So before that person reaches to the adolescent or adult age. If possible, you can also cure it at the very younger age. That is another one important thing. Then you can also determine whether the person is having AIDS or not by comparing the bands of HIV with the person that you have taken. So that is one way. Then the breeding programs. So like you can also go up with evaluate the phenotype of a plant and animal, as it is difficult to make out the homozygous and heterozygous dominancy. Like you have a plant and animal, you cannot see know like the gene that you are getting, whether it is homozygous or heterozygous dominancy, right? Something. So by using this DNA fingerprinting, it will allow to determine the individual phenotype and uses in how can that be used in a breeding race horses and hunting dogs and all. So like if you are clear with it, you can take up the dominant gene one, something like that, the dominant one, and later you can cross over so that in next generation you will get the best out of what you want. So those are the advantages of going with this gel electrophoresis technique. So this was all about applications of human genome project and also extension of it like gel electrophoresis, something like that. Okay. So this was all about today's video lecture. You can, if you are still finding any doubts, you can reach me out through the comment section, or else you can also join the Telegram channel. So where I'm available for you guys. And very important thing is, with this week and even coming next week, this Allied plan and NTA PYQ, the price is also very much affordable price. So if you are planning to, purchase the Allied plan, you can purchase the Allied plan as the price is very much, lesser when compared to the previous one. Like you are getting offers for it. So yes, that is one thing. And do not forget to, solve more number of MCQs, PYQs, and also go through the textbook clearly. Thank you.